This is Autoimmunity, a podcast about our body's never-ending fight with the outside world.
Okay, NPR voice on. Welcome to Autoimmunity episode 44. I'm Kate Franz, and joining me today is Kevin Bonham.
Hello, hello.
Hey, Kevin, and I guess right at the top, just a couple of more housekeeping. We're expanding our digital footprint, improving our digital footprint. Our website, autoimmunity.org. Nope.
Kate.
Autoimmunity.org.
It's
I
been
lost it.
12 years.
Okay, I went in for the NPR and I completely lost it. The problem is we are talking about autoimmunity. I talk about autoimmunity all day long. So that is not what this is. That's a different website, but autoimmunity.org. We have a new website. It looks beautiful. You can check it out. Our episodes are there. Links to support us on Patreon, our YouTube, and our contact is also there. So we have a new contact email, which is contact@autoimmunity.org, and I'll spell it. So contact@AUDIOMMUNITY.org. And did I have the website pulled out in front of me this entire time? Yes, I did. Did it help? No, it did not. You can keep that in.
And if you're joining us, if you're new to the Autoimmunity feed, which you may be because I sent an email into Twiv to try to convert them to Buddhism, and also just to remind people that hey, we're back because it's been like 10 years. And so if you're new, welcome. Matt, who's our other co-host, is unfortunately not able to join us today. But I hope that you will enjoy this paper that we're doing today, which is called Expression Level of Anti-Phage Defense Systems Controls a Trade-Off Between Protection Range and Autoimmunity. And the first author here is Nitzan Aframian, and the last author is Avigdor Eldar, and these are from the Shumnas School of Biomedicine and Cancer Research at Tel Aviv University in Israel. Oh, and it says... there's two co-first authors, I guess. So the other co-first author is Shira Omar Ben-Dori. So this paper is a little bit odd, I think, for autoimmunity, because even though it says autoimmunity in the title, it's about bacteria and phage defense. And normally we think of autoimmunity as being sort of a feature of the adaptive immune system of animals, essentially, of specifically animals that have T cells and B cells and all of the things that we tend to talk about here. This paper, I sort of loved the title and wanted to present it because of that, and this sort of I think blends the way that people normally talk about autoimmunity, and sort of does this fun sort of category bashing thing. But I think it may be confusing to anyone that sort of normally thinks about like T cells and B cells as being the source of autoimmunity. What did you think when you saw that?
I think that if you're coming from like a mammalian or human-centric view, the idea is like, how could this be autoimmunity? We're talking about a single cellular organism where the immune system is made up of millions, billions of cells, hundreds of billions. There's a lot of cells in it.
Yeah.
And so the idea of, some sort of immune reaction, even being, coming from a bacteria, I think is maybe odd to a casual listener, someone who casually follows science. But I think in the last 10 years, and a Nobel Prize would argue that, single cell organisms do have these immune systems. As we know from human and mammalian immunology, there's a very tight balance that you need to be striking between fighting off infection, fighting off cancer, and then protecting yourself from the negative impacts of these cellular responses, which can be like very 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 lethal, right? Like one of the
Yeah.
reasons why we have like this very finely tuned self non-self system is because the self and the non-self can look very similar. And there can be some, mimicry there. So taking that concept into bacteria, into the bacterial immune system, as we call it, I think once we like say what it is, we're
Yeah.
talking, it'll be more obvious. But yeah, I thought that it's, I would say in the best way, this is like a compliment, great
Yeah.
marketing.
Yeah. Oh, totally.
Yeah, like
100%.
yeah, that is not to say it is only marketing, but I appreciated
Well,
the
I mean, it
marketing
worked
of
on
it.
me. It
Yeah,
totally worked on
exactly.
me. And so I thought maybe it might be useful to just sort of back up a sec and talk conceptually about some of the things. I mean, all of this stuff has come up on the podcast before, but there's sort of, a bunch of different ways that we subdivide immune responses. So one, talking again about mammalian systems, we talk about innate versus adaptive immunity, where innate is germline encoded. In other words, it is present in our genomes from the beginning, and the things that are germline encoded tend to target microbial products that are hard for microbes to evolve away from, because if you're going to spend your evolutionary energy, like selective pressure, on something, if it was easy for the microbe to just get rid of it, it wouldn't be a useful strategy long term, because the microbe would evolve faster than we can. They also tend to be things that are sort of structural components, or things where there's a lot of copies of them, so they're sort of lots of available targets. They tend to be present at most phases of a microbe's life cycle. Things like LPS for gram-negative bacteria. LPS, huge component of microbial cell walls. It's always there. There's tons of it. Very good target. Other things that we tend to target are like genomes, so microbial RNA and DNA. Things like that are good targets for the innate immune system, the germline encoded part of the immune system. And then as chordates, is that how you pronounce that? Organisms
Oh,
that we
sure.
normally
Yeah, yeah.
think of as having adaptive immune systems, so T cells
Yeah.
and B cells. Those components of the immune system we call adaptive because they have these weird receptors that get modified during the course of your life. They're sort of randomized. They can target huge ranges of things, and they're specifically selected based on the actual antigens that you encounter. That's the adaptive immune system. We also think, so that's innate versus adaptive.
I
It
like to
turns
think
out,
of it as like the innate is going after the structure. If you think about your house, I can change the things that are inside of my house, but I can't change the bones of the house,
right,
right? And so like, and then the adaptive
depends
is
on how
more
much you want to
saying,
spend on remodeling, but yeah.
okay, well, I can't change the bones of my house. But like I can change, but like if the immune system was just like, I'm gonna go after anyone who has like turkey in their fridge, right? Like there's like some like, something that's, I could just throw the turkey out, but I can't throw out my refrigerator. I can't change my house. But then the adaptive is like learning. Okay, where are you? Okay, I'm gonna like map out where to find you, and I'm gonna like actually travel the streets to your house.
Yeah.
So I don't know; those don't really align. The analogy broke down pretty quickly, but for the innate, I
Well,
think it's there.
another analogy I've used is like if you want to look for cars, you can look for large things with four wheels. But if you're trying to find like a particular, like
Yeah,
someone
yeah,
kidnapped
yeah.
a kid and you're looking for that person, like you need to target a specific license plate, a specific make and model, specific color, etc. So it's sort of the general versus the specific, and it's crucially the way we normally think about things is that to have autoimmunity, to have something that is going to target by accident yourself, it kind of needs to be adaptive, because you wouldn't evolve a innate receptor that would target self. That's sort of like a concept, but at the same time, like we know that innate immune receptors that target microbial genomes, RNA or DNA, can also target self genomes. There's a bunch of systems in place to sort of try to block that. We also know that like a lot of things that we sort of typically think of as adaptive immune responses, there are like more innate like B cells and T cells, and there are more sort of adaptive responses coming from what we normally think of as innate immune cells. So all of these boundaries are fuzzy. This is the Buddhism thing that I keep coming back to. Like categories, models are useful in certain circumstances to sort of help make conceptual sense of these ideas, but all of these boundaries are fuzzy. Evolution will just do whatever the hell works. And this is like a general theme that when we sort of make a model, we make these rigid categories, and then we think that those models describe reality, we often miss a lot of cool biology. And so I feel like this paper sort of does that in a similar way, where getting to bacteria immune systems, so we've known for a long time that bacteria have things like restriction enzymes that will cut certain sequences of DNA. You can sort of think of those as innate-like immune responses, where they'll cut certain sequences of DNA, and then they've evolved, of course, to not have those sequences of DNA in their genome, or they modify their genome in a way that the restriction enzymes don't target. So things like DNA methylation and all kinds of other chemical modifications can sort of protect the bacterial genome. And for a long time, people sort of thought, okay, there's these innate systems for host defense against primarily phages. So phages are viruses that infect bacteria. That's their primary sort of pathogen threat. And we sort of thought, okay, there's these innate systems, and that's what microbes have because they're simple and basic, and they don't get all the fancy, cool adaptive stuff. But then, of course, fairly recently, there's been a lot of excitement around CRISPR and CRISPR-Cas9 sort of systems, and these are essentially adaptive responses. So these systems are generic systems that will target DNA, but they can take information from previous infections and incorporate them in so that this system is kind of adaptive.
I would say it's adaptive, right? It like takes the information, it logs it in its genome, and then it can pull it back out. And then, I mean, maybe it's not as sophisticated as like an adaptive system the way we think about it in the chordates and in humans and mammalian systems, but I think in terms of like how it's actually triggered, right? But I think the general principle of like, here's like a very specific piece I'm going to target. I have encountered it before. I have made some sort of copy or map or receptor to find it, and like I can pull it out at will for subsequent encounters.
Yeah, I think that's right. And I think the only reason I'm saying adaptive like, or I'm sort of hesitating, is because I think that it is even clearer in the case of bacteria that all of these systems have a little bit of adaptive quality and a little bit of innate quality. Gotcha. And it's like all of these systems are under the same selective pressure, which is we want to do the best we can to block or limit the damage of an infection while not going overboard and causing too much damage to ourselves. And like when we think about innate immune receptors, right? So in mammalian systems, TLR9 targets DNA, and that could lead to lots of really dangerous inflammation. So it's tends to be contained within endosomes. In other words, like certain cell types that engulf external material, so that they're waiting for being in a place where a microbe might be broken down and expose their DNA to this receptor. There were early experiments with TLR9 that showed that if you express TLR9 at the cell surface, you'd cause all kinds of inflammation and you'd induce autoimmune disorders and stuff like this. So like clearly the innate immune systems are capable of self-targeting. They're capable of triggering damaging inflammation. And all of the regulation, whether it's the regulation that goes into selecting only certain B cell and T cell clones, or it's the sort of longer term evolution of keeping membrane bound receptors in places where they're not likely to encounter self-antigen, all of that is trying to strike the same balance. We need defense against pathogens, and we need to protect ourselves from damage that the immune system can cause. And the same thing is true in bacteria. It's the exact same selective forces, just on a different scale and targeting different kinds of pathogens, essentially. Does that make sense?
Yeah.
I feel like I'm sort of going... I have all these like thoughts in my head about this and trying to explicate this is sort of...
Yeah. I would say like the pieces that I heard are, like, just to sum up sort of where we're at, is that like humans, like mammalians, bacteria have an immune system that allows them to fight infection. All immune systems, have these complicated and very important regulations in order to strike this balance between fighting an infection and actually detecting and fighting your own tissue. And so when we have that trade-off skewed towards, like if that trade-off becomes skewed, you either go into having, you can have an immune system that like doesn't respond, or you can have this immune system that is overactive and like really can hurt the host.
Yeah.
And I think like the other piece that we're sort of layering over all of this is this idea of like adaptive versus innate systems. And I mean, I think essentially the idea here is we like to think of them as separate. We like to think of the adaptive as the mechanism behind autoimmunity. But in reality, like, these are not separate systems. These are systems that are completely integrated. We normally think of them as moving through one way from innate activation of the adaptive to then fight infection, but it can go back and forth, and you can have these signals and regulation or dysregulation really occur from either side, and they can all lead to these responses that are harmful. And so as that happens for us, it also happens in bacteria. That would be like my high level sort of summary going into this.
Exactly. And so in this paper, they're looking at a class of anti-phage defense systems. So bacteria, a bunch of anti-phage defense systems have been found in lots of different bacteria. Many of them sort of have a particular range of protection. So there's lots of different kinds of phages. Certain kinds of phages are more prevalent in certain kinds of bacteria. And so they're looking at a bunch of different phage systems. They sort of focus on a couple. But one thing I wanted to highlight, they sort of mentioned in the introduction, is that when discovering a new phage defense system, a common practice is to see, like, what is the range of that phage defense system. So you take the phage defense system in a bacteria that you just found, and you, first of all, you like put it into other hosts that get targeted by other phages and see, does this protect against other phages? And this is sort of a common thing that you've done. And one of the notes that they made, which I really appreciated, is they used the word anecdotally to sort of describe like why they started off on this. But it says, anecdotally, mechanism-independent factors such as temperature, multiplicity of infection, so how much phage you give, and expression level have been found to affect the protection range. So you sort of have your standard assays. You're like infecting with so much phage. You express the anti-phage defense system at some level, and you do your assays. But they're saying, well, it's sort of like people know that all of these other factors, not just the specific biochemistry of this gene or set of genes and the phage, all of these other factors can have an effect on how broadly this protection applies. And so that basic observation that there's this anecdote, like, yeah, well, if we like tweak the multiplicity of infection, like it sort of changes the results, or like we change the expression level, we use a different plasmid to express it, we get different results. That anecdote led to this sort of systematic investigation of this property and this whole paper, essentially. So that's sort of where this process started. And so what they want to do is they want to look at whether specifically the level of expression will change the defense range, how many different classes of phages can be protected against, and then they're going to sort of see like if that has additional trade-offs, because if expression level does matter, why has evolution selected for a specific level of expression? And so that's sort of where this gets started. Couple of other things they mentioned. These are just things that I learned because I don't tend to think about phages in bacterial defense. But they mentioned that there's different kinds of ways that the phages can get around defense systems. So one is evasion, which means that they, like you can imagine if a defense system is targeting some particular structure, you might mutate that structure in a way that makes them no longer able to bind. So that's evasion or neutralization. So this happens in a lot of animal pathogens. You produce some factor that targets and disrupts the specific defense. And so both of those might have different strategies of virus evasion as well as responses from the bacteria side to overcome whatever ways that the pathogen is trying to get away from it. So, and so the last sentence of their abstract is the trade-off between protection and autoimmunity may shape the evolution of regulatory strategies and favor the coexistence of multiple systems within a single genome. So that's like a great encapsulation of this whole problem, right? So getting into the paper here, the first figure,
I do just want to hit one small piece. So the way that I understand this is essentially, they increase the expression level of the defense system, and they say, okay, this is going to essentially modify the kinetics and of this chemical reaction. Essentially, this is
right.
Basically enzymatic reactions. We're going to shift the equilibrium. We're going to be able to target more things. What are the outcomes of that? And I think that this is an interesting idea as well. If you think of human immune systems, what would be an analogous way? And I think an analogous way would be to say, okay, we're going to make sure that all of our T cells actually, their T cell receptor, the TCR, actually can see multiple things. So it's going to be cross-reactive to not just one protein in flu, but it's going to hit every single RNA virus entry protein. And what would be the outcome of that? And we can come back to this at the end because these do have some known outcomes of cross-reactivity and what are some of those pieces. But I want to just lay that down, and we can come back at the end of the bacteria to see how different would this be between human and bacterial immune systems.
Yeah, totally. All right, so the first system that they're looking at, they start with this anti-phage defense system that's called SPBK. And SPBK is from Bacteroides. I think I should have actually checked more about where this is coming from. But it's a model organism that has this SPBK. And this anti-phage defense system specifically targets a phage called SP beta, which is a natural pathogen of this particular bacterium. And so what they do is they show if you knock out the SPBK in this bug, you obviously, when you infect with this virus, you get lots of bacterial death. The virus doesn't grow as well. Then they put the SPBK onto a IPTG inducible promoter, which means they can sort of tweak the expression level, and they shoot for a sort of wild type level expression. And you can see that when you have this, you get protection now against the phage. And they try a couple of other phages, one of which, when you have the wild type SPBK expression level, you get protection. But if you have this inducible one and you sort of induce it at a higher level, you get more growth restriction. So the outcome of this phage protection, this part I found a little confusing because when you see a decrease in bacterial growth in these assays, one explanation is the phage is killing a lot of the bacteria. The other explanation is that the phage defense system is working, and so you are getting growth restriction, which is the sort of purpose of this thing. So when you induce the SBBK in the presence of the theta 3T phage, wild type expression level the bacteria grows like normal, so it's successfully defending. But if you overinduce it, you get a reduction in growth. And then if you have the P14 phage, you basically get you don't get any growth restriction, so it's not actually working. I think that's right. No, this is platform immunity. I'm sorry. I'm looking at the wrong. So in Figure 1b, they're looking at the amount of virus. And so in the P14 phage, basically the virus just grows to very high levels, and it's not protecting, even when you overinduce with IPTG. So then they show, because the sort of endpoint analysis is a little hard to understand what's going on, so now they show growth curves, optical density of the actual bugs themselves, and you can sort of see a couple of different patterns. So one pattern, if the microbe is completely protected, you see just like normal growth. The bacteria grow sort of exponential rate. When you have sort of intermediate protection, you get a slowdown initially, but then the bacteria ends up being able to reach its potential. So that's the idea there is you're getting growth restriction in the virus-infected bugs, and that growth restriction is enough to prevent the virus from continuing to replicate and expand, so that eventually the population is then able to recover and grow to its full extent, or you get complete population collapse because the virus is taking over and killing everything, right? So depending on how the protection is, you're either protecting and inhibiting growth, limiting the infection, and then allowing ultimate growth to continue, or you get population collapse because you don't get protection. And they show then in Figure F a bunch of different phages at a bunch of different expression levels, which they're, again, controlling with IPTG. And they show that for some bugs, so for the wild-type bug and the wild-type pathogen, under normal expression levels, you get protection. So that they're sort of filling in this heat map to show the level of protection based on the ability of the population to recover after growth restriction. And you can see that at 30 micromolar of IPTG, which is the sort of high end of induction that they're using in this experiment, that this bug is protected against all but 2 of the phages that they tested, with a couple of different from a couple of different classes. And then now I'm trying to remember what G shows. So that's that's showing. So go ahead.
Oh, I was just going to say it's showing the amount of inhibition of the particular phage, right?
Right. So panel F is showing sort of classification. Like, do you get complete protection? Do you get inhibition followed by protection and eventual recovery? Or do you get complete suppression? And they sort of show these in a sort of binary level. And then in G, they're sort of showing on a continuous scale how much protection you're getting. At 30 micromolar IPCG, there's a range of protection levels, but all of these different phages, there's at least some protection against all of them. Right. So then now they're going to start to get into some molecular experiments. So they take this. So they mentioned that the hypothesis is that this particular phage defense system, the SBBK, is targeting a protein on the phage called a portal protein. So I didn't go and look up phage biology and structure. I assume that it's some part of the tail where the genome is like being inserted into the microbe. Do you know phage biology enough to say what a portal protein is?
I do not know, but let's see here. We could do a quick quick Google.
Okay, while you're doing that, what they did was they said, okay, there's one of the other phages. Okay, so in wild type, all right, I'm just going to step through each of these pieces.
Okay.
All right, coming back. Did you find out what the portal protein?
Oh boy, I was like, this is a bit more.
Do a deep dive.
I was quickly Googling this, and so I was like, okay, this looks correct in that it is important for the DNA packaging of the capsid, but then so when the actual So when phages are forming within the bacteria, they are making their proteins, and they have just a couple of proteins that help make the structure of the phage. So you have a coat protein, a scaffolding protein, and a portal protein. These are some of the ones that you can have. And
Oh, I
the
see.
portal protein seems to be the start of nucleation for the actual like capsid to
Oh,
form.
interesting. Okay.
And then it also interacts with a complex that helps package the DNA. So there's actually a lot of force that goes into packaging up the DNA appropriately in a phage. And some machinery. I think this is essentially the place where the DNA enters and exits from the capsid of the bacteria phage.
Okay.
So
Okay.
it's probably really analogous to like other DNA viruses, like herpes viruses, that also have these really tight, highly pressurized capsids of DNA, and then the portal protein is a place where it pops out, and then
And gets fed in also.
and
during
gets
the
fed in.
Okay, cool. All right, so coming back here to figure 1H. So their hypothesis here is that this other portal protein, even though it has very low sequence homology, is also being targeted when you have sufficient expression of SBBK. And so here what they show, first they show no infection, and so you see sort of exponential growth of the bacteria. If you infect with the theta 29 phage, you start with exponential growth, but then you get complete collapse because the phage is taking over. Then if you have the inducible SBBK and you give it that 30 micromolar, now you see this phenotype where the bugs have this growth inhibition and they have a little bit of population collapse, but eventually they're able to recover and keep growing. So this is an indication that the defense protein is recognizing when you have the high expression level, the defense protein is able to recognize the phage, have growth restriction, limit infection, and then allow the population to eventually recover. Next, they show.
Can I talk about something here?
Yeah, sure.
Do you find it intriguing that they always have to take a hit to their fitness in order to fight off these viruses? So, like, there's always a fitness reduction and then a recovery, versus, I think, for us, I mean, fitness, constant S, it's a very odd, very abstract concept, but the idea that we would take huge fitness costs every time our immune system was activated would be devastating.
Yeah, well,
I don't want to I don't want to draw too tight of an analogy between the immune systems, but it is intriguing that they always have to take a fitness cost.
Right? Because well, so I have a couple of thoughts. So one thing the thing that I struggled with a lot here is the fact that this feels a little bit like population selection. Mm-hmm. So this is a tangent, I suppose, but there's this selfish gene hypothesis that basically selection happens on the unit of an individual, mm-hmm. and that you can't really have population level selection, because it is sort of nonsensical that at a population level you would have individuals that would basically be willing, in quotes from a selective standpoint, to sacrifice themselves for the good of the population. But here it feels like this is pretty clearly refuting that, because individuals are going to take this growth restriction to block infection, even if that individual cell dies. In the concept of an organism, like, we have cells that commit apoptosis all the time, mm-hmm. to protect the organism as a whole, but that's because those individual cells are in service of the germline, essentially. But whereas with a bacterial population, every individual microbe is germline. That said, I feel like because this is a clonal population, that there could be some group level selection where you have, if you are a microbe where your genome contains this defense protein that would allow individual offspring to kill themselves to limit the growth of the pathogen. Theoretically, it seems like that should work at a population level, but I just was a little confused by that. I'm not totally sure. To your point, taking the fitness cost, well, I feel like it's related because, like, yes, taking a fitness cost every time you encounter a virus is bad. On the other hand, if you're taking that fitness cost for the purpose of limiting the infection to all of your genetically nearly identical offspring, like, that seems worth the cost, right?
Yeah. It's different stakes, right?
Yeah.
Like, yeah, because like it's hard. Again, this is why I was saying I don't want to draw the analogy too tightly, but it's interesting to me that you might be evolving a probability, right? And that overall this probability is beneficial for like this culture of bacteria in their survival, but for any one bacteria, there is not a 40% alive. It's binary.
Right.
So it's just, it's interesting to see that like these binary events happening at the level of individual bacteria are actually evolving into this probability across the entire culture and the entire population. I find that intriguing. And
totally.
that is where like the evolution of this has settled.
Yeah, and you can also sort of think of it in terms of like, I mean, there's a whole literature in ecology on cheating, right? So there's all these behaviors. So like you could imagine thinking about a population where everybody has this sort of like group level, like they're going to take one for the team and restrict their own growth to limit the spread of infection. You could imagine any individual bug that got a mutation that sort of like tweaked their growth inhibition back a little bit would be more likely to succeed sort of in particular competitive scenarios, but lineages of that microbe would then maybe be less likely to survive because those lineages, those colonies, those groups of bugs are now more susceptible to a phage invading and killing them off.
Yeah.
So,
and then just because of the replication time, we can see these decisions play out over generations.
yeah,
It's just
exactly.
evolution, hell of a drug. Like it's very, very cool to be able to see like the concept play out on a population level in a way that like you just can't in larger animals or longer generation time organisms.
Yeah, absolutely. Yes. I was going to make some joke about intelligent design and like there's some relation to Darwin's quip about God like really loving beetles and just thinking about, but anyway,
I don't think
I couldn't quite complete that thought because I'm trying to dig into this, but yes.
No, because science is crashing down around us, and the idea of making an intelligent design joke is not funny because it's not ironic anymore, or it's not ironic again. I thought we like moved past this.
Yeah. No, that's fair. That's fair. Yeah. Just
anyway.
Side note to say, we're recording on August 7th, 2025, in a time the week after our Secretary of Health and Human Services, who evidently other reporting suggests that he doesn't believe in germ theory, that our Secretary of Health and Human Services just like canceled, I forget what it was, $500 million of funding for mRNA vaccines, which are like one of the greatest public health inventions of the last 50 years. Like, I don't know if it's quite on the level of antibiotics, but I feel like it could approach that level of impact. Just the ability to create a vaccine against almost anything, almost instantly. Anyway.
Yeah,
little aside,
from like a national preparedness perspective, they're phenomenal tools, and it just seems foolish to not use every tool available to you, and to decide that certain technologies are in favor and other ones are not off of the whim and not off of the scientific mettle of the approach and the safety of the approach and the ability for a government to respond to a pandemic. It seems pretty foolish. And then on top of the fact that I think we're about to see just like a revolution of genetic medicines and mRNAs. Like I'm not, there you always look silly maybe in the from the future saying like this is going to revolutionize, so I don't want to like oversell it, but that is to say you don't always know what's going to be so impactful, and you don't know that until you continue to fund the research and figure out specifically what are the use cases and what are the broader impacts going to be. This is one of those things that from the outset looks like it's already done a lot of really great things. It's going to continue to do great things.
I'm laughing because one of the reasons I picked this paper is because I wanted to get away from the like politics, and it feels like phage and bacteria host defense. Like it's not we don't have to talk about animal models. We don't have to talk about all of this other stuff, and yet this is just in the air right now. So if you're listening to this
in the future,
in 5 or 10 years,
and everything is fine.
Either everything is fine, or you are discovering this in a post-apocalyptic hellscape. Just know that in late 2025,
we were losing our minds.
Scientists were suffering. We were losing our minds. Yeah, that's a good way of putting it. We were losing our minds.
Yeah.
Either way, it turns out.
You're listening to the post-consolidation, and there are no more independent academic labs, and there are no more small startup biotechs, and actually academics is just a private-public partnership between AbbVie and Stanford, and we only train 20 scientists a year, and then we just hire hands in order to do experiments, to make data, to feed the AI. If that's where you are right now, I'm sure I'm really loving my AI master. And also, I never doubted you for a second. Can I please have the 4th of July off?
Yeah. Yeah.
Maybe we wrap this up before we.
What's good in science in 2025 is Matt's phrase. Coming back to this. Okay. So they then show a wild type. So they have what they call an escaper mutant now. So a bug or a phage that has this portal protein, but it has a mutation that escapes the normal phage defense against it. And basically, when they infect wild type bugs with normal expression levels of SBBK, they get more growth restriction, a deeper dip, so basically the infection is worse, and then it doesn't recover. It recovers, but not quite as well, and they show that if they overproduce SBBK, it's essentially the same phenotype. In other words, you still have a lot of this growth restriction. It's still very hard for the bug to fight it off. Right? So that's looking at infection. So in the next experiment, this confused me for a very long time. But in the next experiment, basically they're just overexpressing, they're not infecting with a phage, they're overexpressing this portal protein in the microbe to just see the effects of this interaction directly. Basically to separate out the growth restriction from the destruction by the virus itself, right? The lytic properties of the phage. And basically they show the same thing. So you when you overexpress the SBBK, or actually no, they don't have overexpression here. They show that if you in the presence of SBBK wild type and Yon-E, which is the original phages thing, you have a bunch of growth restriction. If you put in the wild type theta 29 GP10 protein, you have some growth restriction. If you put in the escaper mutant, you don't have any growth restriction at all. And then if you put in the theta 29 in the absence of SBBK, you also don't have any growth restriction. So this is just sort of confirming that indeed the interaction of SBBK with the portal protein of either the original phage or this theta 29 phage, both of those portal proteins both respond and induce growth restriction in this bug. Okay. So what they've established now is that you can increase the host range of this phage defense system by just overexpressing it, by increasing the amount. And they're starting to show a little bit that maybe this is having some consequences in terms of fitness of the bug. And so in figure 2, they basically want to say, like, is this is this just a property of this specific phage defense system, or is it a general property? And so they show with a bunch of different phage defense systems and this same sort of list of phages that we have a mix where in many cases, if you overexpress, you get defense that is expanded compared to being at native expression levels. And then for one of these specifically, so all of those were in B. subtilis. That's the bug I kept forgetting. Model organism Bacteroides subtilis? No.
Bacillus.
Bacteroides?
Bacillus.
I always say Bacillus subtilis. Thank you. Yeah, I thought that was wrong. We always just say B. subtilis. Bacillus subtilis. Thank you. And then so they showed this again in E. coli, a different group of phages and a specific E. coli defense protein. Again, you have a mix of defense at native expression, defense at high expression, and then improved defense at high expression. And these phages that they're looking at have a mix of the sort of evasion strategy versus neutralization strategies. And you have similar sort of similar behaviors. And so now what they want to do is they want to say, like, is overexpression able to overcome neutralization? Is it able to overcome evasion, or both? And basically what they show is that if you have something that is evading or neutralizing or both, you need increasing levels of expression of the phage defense system in order to get protection. So you need the most expression to overcome when they are doing both neutralization and evasion, which kind of makes sense. I was actually surprised. I would have expected evasion to be harder to overcome with overexpression. Because if you're just avoiding being seen entirely, that's different than if you're trying to produce something that is neutralizing and then you just overexpress the agent, and so you can't neutralize it as well. But I guess the evasion is only partial, and so if you have someone that's sneaky, you send more cops out looking for them, you're more likely to find them anyway.
I will say the piece that, and this is clouded by like how well I understand these systems, which is not at all. But from the idea of evasion,
I'm just going to talk out of my ass. Excuse me.
No. I'm just going to give like a biologically, chemically informed opinion on. Yes. It depends on what the triggering the protection does. If you only need to trigger one defense system protein, if like the protein that has evolved in such a way to evade like detection, as you increase the amount of protein defense present, you will have non-specific interactions that will be able to trigger the defense. If your defense requires every single protein to be like that enzymatic reaction to occur like perfectly, no, like the evasion will be effective. But if you have to evade everything, when you increase the concentration of that defense protein, you will increase the idea that maybe it just self-activates, right? Like there are different ways that these proteins, like how, all that's to say, the mechanism is really going to define how effective overexpression is for evasion. Yeah, absolutely. All right. So now they've sort of said, okay, for this one phage system, overexpression seems to increase range. In figure 2, they say across a broad range of phage and defense systems, we are seeing a similar pattern. So if it were the case that we could just increase expression and get added protection, why wouldn't the microbes do that, right? And they mention, I thought a nice little flourish in the paper. I can't remember where it was, but they said basically like increasing the expression of a protein is like a pretty easy mutation, like easy, like it is easy to increase the expression of something like a promoter change or something. Like it's not like you need to like invent a whole new structure in evolution. It's just like minor changes can change expression. And so you would think that if this is an available defense, go ahead. Oh yeah, sorry. I I think I'm about to say exactly what you're going to say, which is that the bacteria will explore the mutational space to increase the expression. There's no way that the bacteria haven't had a mutation to overexpress this protein. Why isn't it protective?
Yeah,
Like,
exactly.
just in this experiment alone, this group probably did enough infections, enough bacteria to be able to isolate a bacteria with this mutation in and of itself. If they had to like sequence these populations, someone has it. Why isn't it the dominant genotype?
Yeah, exactly. Yeah. If this was an available defense, why wouldn't they use it?
Yeah.
To increase their range. And so then they set out to test, like, are there downsides to overexpression? And they do this again across a range of defense systems. So in panel A of figure 3, the first sort of plot they're showing here is this SPBK. And they show that up to 30 micromolar of IPTG induction, which is what they used in previous experiments, you actually don't get much growth restriction at growing at 37, which is panel A. But if you go above that, if you go 50 micromolar or 100 micromolar, all of a sudden now you're getting pretty strong growth inhibition of in these bugs. So that's a suggestion that, okay, well, maybe we could get up to 30 micromolar. We could get up to this level of increase, which, again, increased the protective range quite a lot. But above that, you're getting growth restriction. So, maybe that's a problem. They show a bunch of other systems. Some of them are IPTG inducible. Some of them are inducible with other sugars. So this is just, I was confused for a moment. I was like, xylose, where did that come from? But there's just different ways of overexpressing genes depending on the phage defense system. I'm guessing it was just like what plasmids they had available or whatever. And in all these other ones, they can do pretty high levels of expression and not get growth inhibition. So it's like, well, okay, so this is maybe available. But then they show in panel B, if you decrease the temperature of growth, now there's sort of more restriction even at the lower expression. So this is, if you're in optimal conditions, right? They're growing these bugs in like rich media. These bugs are as happy as they could possibly be at 37 degrees. So maybe under those conditions, yeah, you can just overexpress the crap out of these things and it's still fine. But now if you start to add any kind of other adversity, all of a sudden overexpression becomes a little bit more of a problem.
Yeah, so you've really lost their adaptability. Quick question.
Yeah.
What figure data was completed first? Figure 2 or figure 3? Because figure 3 to me reads, I've made this inducible system. I'm now going to test it. Blah, blah,
in
blah,
every
blah,
way
blah.
possible.
And then they went, oh wow, interesting. When I induce it to this really high amount, all my bacteria are dying. This isn't good.
Yeah.
I wonder if figure 3A, the upper left-hand panel, is the genesis of the paper. To me, this feels like the finding that sets you down the road of this paper.
That seems entirely plausible to me. Yeah, I would believe that.
Anyhow.
I mean, we should reach out to these authors and ask them this kind of thing.
I mean, because we definitely are getting this wrong.
It is. Sometimes it is more fun to just wildly speculate about possibilities than it is to actually know the truth. So don't come after me. So then they do what I think is sort of the next. I think the next obvious experiment here, if you're going to make the claim that there's some selective pressure, even with tons of overexpression, you're not seeing growth defects. So what's another way that you can look at potential selective pressure? By doing competition experiments. So now what they're going to do is they're going to put in co-culture bugs where they can induce high levels of this versus bugs that are maintaining wild type. And effectively what they show is that across many of these, even when you didn't see a growth inhibition at very high expression levels, if you are culturing that same bug with another bug that doesn't overexpress the defense system, you are at a competitive disadvantage. So again, if you had external adversity, whether in the form of decreased temperature, I would suspect if you put in like nutrient restriction, if you did like minimal media or something, you might see a similar effect. And also, if you have competition, even from the exact same species, just with less expression, you also have a selective disadvantage in this presence, or in the presence of overexpression of these defense systems.
Sorry, when you say competition of the bug, these are competition of the phages? These are two different phages, a mutant and a wild type phage at induced and uninduced.
No,
with induced and
I
uninduced.
think
Sorry,
it's just the bacteria. So they're co-culturing like a bug where you can do IPTG induction of the defense system, or a bug where IPTG will not induce overexpression. You put those two bugs together in the absence of phage, just overexpression gives a selective disadvantage in competition.
Gotcha.
With other microbes. I'm like 90% sure that's true.
I think you're correct. Yeah. Sorry,
growth curves of strains carrying either inducible wild type or inducible mutant systems in the absence or presence of an inducer. Yeah, so they also have mutants where like they don't cause the growth restriction, and those do fine. So that's just to show that it's not just the overexpression of the protein. It's not like synthesis of the protein using more resources is the problem. The mutants are saying these are inactive. They can't induce growth restriction like the defense strategy, and we can overexpress them like crazy, and they're still not. That's not a competitive disadvantage. But when you have the protein that can induce growth restriction as part of its function in competition, that causes issues.
Gotcha. Yeah.
And then figure 4 is just a schematic, which feels superfluous, but it's fine. It's a pretty little cartoon. I like all of the different phages with the different size tails and stuff.
Yeah.
But yeah, so this is, I mean, it's a fairly simple paper, I think, conceptually. A lot of work. There's like a ton of different genetic systems. Like, I'm sure that this grad student or postdoc, or I guess these two grad students or postdocs, whoever they are, we're in the lab at all hours doing these like long-term growth experiments, collecting data. But conceptually simple. I think, again, this is a theme of season 2 here of autoimmunity. Oh, now you've got me doing it. Season 2 of autoimmunity, which is that if you have a conceptual model where you are saying something cannot be true because of my conceptual model, maybe you should just look and just see if it's true. Because we've seen now where, immune system in the brain where it shouldn't be because blood-brain barrier. Here we see bacteria with immune systems that are causing autoimmunity to some definition in bacteria. Lots of other examples where if you just go look, you might find something surprising. But conceptually, I think it totally makes sense. This 100% makes sense. If you step back from the sort of narrow definitions and say, conceptually, what would we think would happen with a phage defense system if you overexpressed it? Like, if it was easy to fight off pathogens, there wouldn't be pathogens. So there must be some other reason to restrict it.
Yeah.
So I thought that was pretty cool.
Yeah, I like the concept of this. I'm going to come back to what I opened our discussion of the paper, just how does this really relate to our immune systems? And so we do see that in the course of trying to fight viral infections, we can have T cells that have specificity for viral proteins, or especially during the expansion phase, like that effector phase of the T cells, we will see TCRs that are, trying to fight the infection, but we also get a lot of TCRs that are cross-reactive to host antigens. For a variety of reasons, we might expand those T cells. And it's kind of similar to this idea here of like, if we just expand the amount of cells that can fight these infections, we should be able to fight our infection appropriately, resolve the infection, continue on with our life, with our day. And normally that's fine, having like these autoreactive or, TCRs that like fight viruses, but also might cross-react with human antigen. Because at the end of fighting an infection, we have a contraction of our T cells, right? So that's how we really are able to handle this potential cross-reactivity. Now, in some cases, it might be because of your age, it might be because of just random chance or something genetic. The T cells don't contract as well, and you don't delete some of these, short-lived effector cells, and those cells can then form into your memory pool, and you can have a T cell memory pool that is cross-reactive to your own host antigens, and this is one mechanism of autoimmunity, and so I think like the parallels here. We see like conceptually, we see like this really interesting parallel of, increasing range, increasing antigen specificity, really can have detrimental effects on the host. And so like these are just really tight balance. So that was sort of the concept I was hinting at the beginning.
Totally. Yeah. I think of it as sort of like there's a razor's edge.
Yes.
You lean on one side, you're susceptible to infection. Lean on the other side, you're susceptible to autoinflammation or destruction. And I think, the other thing that's really funny about this is that the pathogens they have a very similar sort of orthogonal evolutionary balancing that they have to do. Where on the one hand, they don't want to replicate so completely out of control that they just completely overwhelm and destroy a host, because then they're not going to be around long enough to spread. Yeah. And so, we often see pathogens, they're not optimizing, they're not evolved for virulence. They're optimized for replication and transmission. Replication pushes towards more virulence because, more replication, using up more host resources means you can make more copies. But if you go too far down that path, then you are not able to transmit as effectively. And so, I've always thought it was so interesting that, flies don't have T cells and B cells, and they do just fine. But if you take away a human's or a mouse's T cells and B cells, they're completely screwed, because the pathogens and the hosts are sort of evolving in this arms race, and if one gets too far ahead, then it's bad for everybody involved.
Yeah.
So
don't tell my rag knockout mice that they're screwed, please.
I mean, one benefit of working with rag knockout mice is if they escape into the building, you're not going to have a problem for very long. Like, those mice are.
You just follow like the little trail of poop, and then you'll find the mouse.
Yeah, exactly. I remember seeing white mice running around the Model Center a bunch in graduate school because people were not handling there. Anyway, totally orthogonal. This has been Autoimmunity, recorded on August 7th. Hopefully Matt will be back with us next time. As Kate said, you should check out our beautiful new website. I used Claude Code to help me with the CSS because I don't care to learn how to style a website. That's at autoimmunity.org, AUDIOMMUNITY.org. There you can find links to our Patreon page, patreon.com/autoimmunity if you want to support us. Ways to contact us, like Kate said, contact at autoimmunity.org. I also made a Blue Sky account, which is currently autoimmunity.bsky.app, but I think I'm going to change that to be, you can verify it with your website, so I'm probably going to change that to be autoimmunity.org, but you can follow us and you'll stick with it even if I change the handle there. What else? You can see all of our old episodes. We're putting stuff on YouTube, but there's also an RSS feed, iTunes. If you leave us a rating and review, that would be great. If you want to kick us $1 or $2 per episode, that would also be great. But really, the best way you can support us is by sharing us with your friends. Tell other people about us. We would love to have more listeners. We would love your feedback and suggestions. Any of those different ways of contacting us. I think that's it. The music at the beginning and the end are composed by Rachel Reineck, and we'll see you next time.
Great. Thank you, guys. Awesome. Okay.
Sometimes you get